I'm Jay Trautman, as was said, co-founder and CEO of Invenio, where we've developed the Neo laser imaging system for real-time imaging of fresh tissue in the OR. It's been used in over 5,000 clinical cases to date, and we're now adding an AI layer for immediate analysis of those images in a 5-center lung study funded in part by the Johnson & Johnson Lung Cancer Initiative and the NIH. So why are we working on lung cancer? It's still the leading cause of cancer-related deaths. To try to reduce those rates, there are screening programs in place, so high-risk patients are CT screened. That results in 1.5 million nodules being identified, which is about 750,000 procedures every year to establish a diagnosis for those patients. But about 20% of those 750,000 procedures have failed biopsies. So a lot of money has been thrown at this problem and has improved the outcome. Along here at the bottom, some of the investments made in the area in robotic bronchoscopes and navigation. But probably the best way to improve the outcome is to examine the biopsy when it's taken with a real-time technology. And so that's what's recommended by US CAP, that a cytologist be present in the procedure and provide a real-time read. But it's not widely available. Those people are hard to hire, and the quality is low, as well as it being labor intensive. So our technology is based on a laser spectroscopy, an imaging method that came out of a lab at Harvard. My co-founder and our CTO is co-inventor of the method. So it allows us to take fresh tissue, it's placed in our consumable and dropped in the instrument, and in a couple of minutes you get a high quality histology image with no processing of the tissue. That tissue is not damaged in any way, and so it can be used after the fact for downstream analysis. Now we're adding the instant AI interpretation, which I'll tell you more about. As mentioned, it's been used in over 5,000 cases. We have 16 systems running around the world right now. That's actually from 12 hospitals. 4 of those hospitals bought second systems to branch out into new disciplines, like the technology so much. And that's without the AI. That's just using it with existing human reads. So it works across all these different sample types. We began in brain because our initial collaborator was a brain tumor surgeon. But as I've indicated, we're focused primarily on lung right now for the AI development. We really strove to get the best possible image quality when we built the system, because that's what drives diagnosis in histology. And so the other technologies that are being used to attack this problem have poorer image quality than ours. And I mean, that was done initially because we were worried about pathologists reading the images, which they of course can, but it also makes a huge difference in the AI. So another feature that you get, because there's no processing of the tissue, is the reproducibility from site to site is just amazing. And so when we've built a number of the AIs, we've got data coming from multiple sites, and we use relatively small data sets to complete the AIs. So these are examples of AIs that we built. Differential diagnosis. Molecular markers is a particularly interesting one where The markers were identified based on the histology in these diffuse glioma samples. Those are both academic collaborations, Nature Medicine publications. What we're focused on is the simple detection of cancer in the lung biopsies for our first FDA clearance. And so the Lung Cancer Initiative, Johnson & Johnson, is supporting the study as well as the National Cancer Institute. Where we stand in the study is we're up and running at the various sites. We spent a good deal of time with the FDA over the last year and a half to get to the point that they were accepting of our endpoint. This is a de novo route because there is no predicate for this technology. So we're moving fairly quickly now through the roughly 1,000 patients from these 5 sites that we need to get clearance of 3 different AIs for the 3 different sample types that are taken in the study procedure. So I think the value is pretty obvious. I mean, for those 150,000 patients, if they didn't get an initial diagnosis, some of those may had to go back for another procedure. Some just were put on a wait list and may have actually had the condition, but it wasn't known. So getting diagnosis for people immediately is a benefit both financially and for the patients psychologically. Where we go next with this is the push into real-time diagnosis, enabling same-day treatments and even same procedure treatments. So we've been exploring that both in lung and at length with one of our collaborators in prostate. So there's quite a market just in the biopsy end of the interventional pulmonology. Moving into procedures will open up even more. Urology is the area that we'll go into next with studies that we'll take to the FDA. There's focus on targeted biopsies. We've already got some interesting data about optimizing ablations as well. So the team is small, even though we produce this. So manufacturing is in Santa Clara, AI is developed in-house, and we run the clinical study. So on top of the non-dilutive monies from Johnson & Johnson and NIH, we need to add some private capital to get all the way through the study and clearance at FDA of the first AI and submission of the second 2 AIs. Late in that process, there are a number of strategics that we're working with, so we'll explore commercialization, with the strategic and alternatively put together a B round for commercialization ourselves, depending on what looks like the best option. Thank you.